How did a single structural protein, shaped by ancient environments and inherited through countless lineages, come to determine the stability of human muscle? This question anchors the story of dystrophin, a molecule whose absence produces the characteristic weakness observed in Duchenne Muscular Dystrophy. Its role is neither dramatic nor visible at first glance. Instead, it reflects a quiet interface between genetic instruction and mechanical force. Today, researchers trace this condition to specific mutations on the X chromosome, examining how small alterations influence muscle fiber behavior across global populations. The pattern illustrates how inheritance, gene flow, and evolutionary history converge around one molecular function that maintains cell integrity under repeated physical stress. The dystrophin gene functions as a structural blueprint for a protein that reinforces muscle cell membranes during repeated mechanical stress. Its size, extending across a substantial region of the X chromosome, increases the likelihood of mutation through replication errors. These changes alter the continuity of the genetic code and interrupt dystrophin production. The mechanism begins with transcription across numerous exons, followed by translation into a long, flexible protein designed to connect internal, cytoskeletal components with surrounding stabilizing structures. When any segment of this sequence is disrupted, the resulting protein becomes absent or incomplete, limiting the ability of muscle fibers to maintain structural integrity. Dystrophin operates within the dystrophin-associated glycoprotein complex, a network that distributes contraction forces across the membrane. This interaction reduces shear stress on individual fibers and stabilizes the lipid bilayer during daily movement. In the absence of dystrophin, the membrane becomes more permeable and susceptible to mechanical injury. Evidence from muscle biopsies and cell models consistently shows increased membrane rupture, ion imbalance, and activation of inflammatory pathways. Over time, repeated damage leads to cycles of degeneration and repair, eventually transitioning toward fibrosis as functional fibers are replaced by connective tissue. Geographically, dystrophin gene variants occur across all major populations. Their distribution reflects mutation drift rather than adaptation, since the protein serves a universal structural role without known climate-driven variation. Ancient DNA has not revealed region-specific selective pressures, supporting the conclusion that dystrophin stability relates primarily to general cellular mechanics rather than environmental contexts. However, differences in diagnostic capacity influence reported prevalence and detection timing. Today, the gene's size, structure, and vulnerability to mutation form the core explanation for Duchenne muscular dystrophy. Its biological role remains consistent across lineages, demonstrating how a single molecular component contributes to muscle behavior, endurance, and stability. This understanding provides the foundation for examining how distinct mutation types influence disease progression. In subsequent chapters, mutations affecting the dystrophin gene fall into several well-defined categories, each altering protein production through specific molecular mechanisms. Deletions remove one or more exons, shifting the reading frame and preventing formation of a functional protein. Insertions or duplications disrupt sequence continuity in similar ways, while point mutations modify individual nucleotides and can prematurely halt translation. These patterns consistently interfere with dystrophin's structural role, limiting its capacity to stabilize muscle membranes during contraction. The functional consequences of these mutations emerge through predictable cellular behaviors. Without full-length dystrophin, the dystrophin-associated glycoprotein complex becomes unstable, reducing its ability to distribute mechanical force across the membrane. Muscle fibers then experience localized stress, creating small tears that allow the uncontrolled entry of calcium ions. This influx activates enzymes that degrade structural components and initiate inflammatory responses. Repeated damage and repair cycles gradually reduce the proportion of viable fibers, contributing to the characteristic decline in muscle strength. Evidence from clinical studies shows that out-of-frame deletions generally produce the most severe outcomes because they eliminate the possibility of generating even a partially functional protein. In-frame deletions, by contrast, may allow production of shorter but still active protein variants, leading to milder phenotypes in related conditions. Molecular diagnostics, including multiplex PCR and next-generation sequencing, have clarified these distinctions and improved identification of specific mutation types. Across global populations, mutation spectra remain broadly similar, suggesting that these alterations arise from universal cellular processes rather than local environmental pressures. However, differences in reported prevalence reflect variation in clinical resources, genetic screening programs, and research infrastructure. Ancient DNA sources rarely preserve intact dystrophin sequences, but available data support the view that mutation patterns follow general demographic expansion in gene flow rather than climate-driven selection. Today, understanding mutation categories provides a structured explanation for the diverse presentations of Duchenne muscular dystrophy. Each variant influences membrane stability, fiber resilience, and long-term muscle behavior, forming the basis for later discussions on disease progression and therapeutic development. The absence of functional dystrophin initiates a predictable sequence of cellular events that steadily undermines muscle integrity. Without adequate linkage between the cytoskeleton and the extracellular matrix, mechanical force becomes unevenly distributed across the membrane. This imbalance produces small disruptions in membrane stability, especially during repeated contraction cycles. These openings permit excessive calcium entry, which activates proteases that degrade essential structural proteins and weaken the fiber's internal framework. As these processes continue, muscle fibers experience increased susceptibility to further mechanical stress. Damaged fibers release intracellular components that attract immune cells, initiating a localized inflammatory response. While this reaction aims to remove compromised tissue, it also contributes to additional stress on surrounding fibers. Satellite cells attempt to repair the damage, but their regenerative capacity becomes strained as cycles of injury and repair accelerate. Over time, the pool of functional satellite cells diminishes, limiting the muscle's ability to maintain structural continuity. Evidence from histological studies consistently demonstrates this progression. Early biopsies show scattered fiber necrosis alongside infiltration of macrophages, while later samples reveal significant replacement of muscle tissue with fat and connective material. The shift toward non-contractile components reduces overall muscle force and accelerates performance decline. This pattern appears across diverse populations, indicating a shared underlying mechanism, independent of climate or environmental pressures. Now, advancements in imaging and molecular assays have provided clearer insights into these pathways. Calcium-sensitive dyes, membrane integrity markers, and transcriptomic profiles all reveal consistent signatures of stress, inflammation, and impaired regeneration. Comparative data from related muscular dystrophies enhance understanding of why dystrophin's absence produces uniquely rapid decline. Today, these mechanistic insights guide therapeutic development, including efforts to stabilize membranes, reduce calcium influx, and enhance regenerative reserves. The pathway from initial membrane instability to widespread fibrosis provides a structured map of the disease's progression and frames the basis for emerging interventions. Early indicators of Duchenne muscular dystrophy emerge gradually, often reflecting subtle deviations in the expected trajectory of motor development. Most observations involve delayed milestones, such as late walking onset or difficulty maintaining balance during basic movements. These behaviors result from the underlying muscle fragility caused by insufficient dystrophin, which compromises force transmission during normal childhood activity. However, early variability among children can delay recognition, making structured diagnostic pathways essential for accurate assessment. Clinical evaluation typically begins with observation of gait patterns, postural stability, and the presence of compensatory movements. One frequently noted sign is the use of the upper body to rise from the floor, a behavior associated with weakened proximal muscles. Laboratory testing often follows, with creatine kinase concentration serving as an initial biochemical indicator of muscle membrane disruption. Elevated levels suggest ongoing fiber damage, although confirmatory tests remain necessary to establish a precise diagnosis. Today, genetic testing provides the most definitive approach to identifying dystrophin gene mutations. Modern sequencing platforms can detect deletions, duplications, or point variants across the gene's extensive coding region. This method clarifies mutation patterns and offers insight into expected progression rates. In many regions, newborn screening programs are being evaluated as potential tools for earlier detection, reflecting the growing recognition of the benefits associated with timely intervention. Imaging techniques complement molecular testing by visualizing early structural changes within muscle tissue. Ultrasound can detect increased echogenicity associated with fatty replacement, while magnetic resonance imaging offers detailed mapping of muscle composition. These modalities help clinicians differentiate Duchenne muscular dystrophy from other neuromuscular conditions with overlapping early features. Now, diagnostic strategies increasingly integrate molecular, biochemical, and imaging data into unified evaluation frameworks. This combined approach improves accuracy, reduces diagnostic delays, and supports earlier planning for therapeutic support. The careful identification of early markers enhances understanding of disease onset and informs broader efforts to refine intervention timing. The transition from childhood into adolescence marks a period when the functional consequences of dystrophin absence become increasingly apparent. During early childhood, muscle weakness progresses gradually, allowing many daily activities to continue with minor adaptations. However, as body mass increases and mechanical demands intensify, the imbalance between muscle resilience and physical load begins to widen. This shift accelerates fatigue, reduces endurance, and amplifies gait abnormalities already present in earlier years. One consistent pattern involves the gradual decline in proximal muscle strength. Particularly in the hip and thigh regions, children often exhibit increased reliance on compensatory strategies, such as widening their stance or shifting weight laterally to stabilize movement. These adaptations help maintain mobility, but also place additional strain on already vulnerable muscle groups. By comparison, upper limb strength generally decreases at a slower rate, although functional limitations emerge as cumulative muscle damage advances. Today, longitudinal studies highlight the predictable progression of structural changes within muscle tissue. MRI scans reveal increasing fatty infiltration during late childhood, followed by widespread replacement of contractile fibers during early adolescence. These shifts correlate with measurable reductions in walking speed, stair climbing ability, and overall motor endurance. In many cases, the loss of independent ambulation occurs during this period, reflecting the combined impact of mechanical stress, inflammation, and declining regenerative capacity. Respiratory and cardiac muscles begin to show subtle signs of strain as well. Reduced pulmonary function becomes detectable through routine monitoring, while early changes in cardiac rhythm or structure may appear during adolescence. These trends provide important markers for clinical planning and highlight the multisystem nature of the condition. Now, the progression across this developmental stage illustrates the interconnected biological pathways that define Duchenne muscular dystrophy. The steady accumulation of structural changes, functional adaptations, and systemic involvement forms a clear continuum that shapes management strategies throughout adolescence.